Welcome back. Last lecture clip here for Chapter 3. We're going to discuss a little bit about cell structures, those internal parts. So I'm bringing back up this figure from Chapter 3, which is a generic cell inside your body. This is also, incidentally, what the inside of the cells of many different living things here on the planet would look like. So a lot of these little structures and their functions and so forth that we're going to talk about here would apply to many other living things as well. All right, so again, we've already spent a lot of time talking about the plasma membrane, that purplish covering they've got depicted on there. And then on the inside of the cell, you have these various structures depicted here, and we'll talk a little bit about their functions. Now, your textbook, again, is going to go into a lot more detail than we are here. We're just going to hit some highlights so that you have a little bit of familiarity with these internal structures for when they do come up at times throughout your A&P coursework. All right, a little terminology. So again, everything that's between the plasma membrane and the nucleus is referred to as the cytoplasm. So that includes the fluid in there, all the dissolved substances, the solutes in the fluid, and then the organelles, these structures that have specialized jobs that we're going to have an overview about. The cytoplasm also can include little storage inclusions or inclusion bodies where things like glycogen, that's a starch for storing glucose sugars, or you can have little lipid droplets like in your fat cells where fats are stored for energy when you need them. Those are called inclusions, little locations inside a cell where they concentrate things. But all of that stuff between the plasma membrane and the nucleus together is called the cytoplasm. This term cytosol, sometimes people are confused about cytosol versus cytoplasm. Cytosol refers to the fluid itself, the water with all of the solutes that occupies that space in there. Probably would have more like a jelly-like consistency than an actual fluid type consistency if you could actually scoop some out and squish it around between your fingers. All right, how about the organelles inside our cells? There are many of those. Many of these, all 200 different cell types, trillions of cells within your body. Many of these they have in common. But then in some cases, some cells don't have some of these, depending on what that cell type is. But in general, most of your cells are going to have these structures, these organelles. And some of them are said to be membranous, all right, and that means they are themselves covered with a phospholipid bilayer membrane, but it's just an internal membrane inside the cell. And then others are non-membranous, which means they don't have a phospholipid bilayer membrane surrounding them. All right, so let's talk about some of our membranous, some of these organelles here. First one, the mitochondria. What are those? If you look on your cell diagram picture, you'll see little things that look like sausages scattered around the inside of the cell. Those are your mitochondria. They actually have a double membrane structure, which winds up being important for their function. Best way to think about mitochondria, these are the powerhouses of your cells. I'm going to change to a darker pen color here. There we go. Those are the locations where a cell makes most of its ATP, and there's a process for that, which we'll talk about in Unit 2, called aerobic cellular respiration. That's a bunch of chemical reactions that take place, and cells perform those reactions to harness energy from nutrients, like the sugars you've taken in from your foods. They capture that energy and they store it in these ATP fuel molecules. And so that process happens in your mitochondria. So just think about your mitochondria, associate them with making ATP fuel, and they're like your power stations for your cells. All right, ribosomes are another type of organelle, and these are non-membranous themselves. They're actually like little granules. They're scattered all throughout the inside of the cell, and they themselves are built from proteins and RNA molecules. The little lowercase r there means ribosomal RNA. So in this, the proteins and the ribosomal RNAs get together and form these little granules. So they're actually the building blocks of the ribosomes themselves. Ribosomes, though, are your sites of protein synthesis. They make proteins. So they read those genetic instructions that we talked a little bit about and interpret those and link amino acids together to build proteins. We'll hear more about that in Unit 2 as well. So yes, they are built from proteins and RNA, but then they are the machines that build your protein. So think about with ribosomes, think protein synthesis. Okay, so many of your ribosomes are what we call free ribosomes. That means that they're floating around in the cytosol, and they make what we call soluble proteins that are dissolved in fluids surrounded by the watery fluids of the cytoplasm. And then others are called membrane-bound ribosomes. Those are attached to, let me back up over here. Didn't realize I was going to have to back up that far. Those membrane-bound ribosomes, see this big blue structure here? This big blue structure here, that's called the endoplasmic reticulum. It's abbreviated as ER. And the rough ER has ribosomes attached to it. And if you're looking at that diagram more closely, you'll see all those little like pinkish red spots there attached to the ER. Those are supposed to be the ribosomes that are attached to it. All right, so the ones that are attached to the ER, they make proteins that are now, we've already seen you have lots of proteins in your cell membranes. So that's where those kinds of proteins are made, because they're not going to float around in the fluids inside the cell. They actually have to be stuck into a membrane, or sometimes cells make proteins that have to be exported by exocytosis, if you remember that term, a type of vesicular transport. And those types of proteins also get made by these ribosomes that are attached to the rough ER inside a cell. All right, so a little bit more about this ER, endoplasmic reticulum. So that is can take up a lot of space inside a cell. It's an interconnected, like a web. That's what when you see reticulum, that's referring to something that's webby in appearance, like a spider web. So these are membrane-covered tubes and compartments that are all connected to each other and have kind of a webby type appearance. And don't worry about cisternae or cisternae for now. You'll hear that again when we get to the muscular system, but don't worry about that for now. The membrane of the ER blends in with the membrane surrounding the nucleus in a cell. And again, there are two varieties. Rough ER, that's the membrane of the rough ER. It has ribosomes attached to it that are building proteins. Smooth ER does not have ribosomes attached to it. And here's a depiction of that. So in purple here, this is supposed to be a portion of the nuclear envelope in purple, and the blue structure you see there is the endoplasmic reticulum. So you can see how it's kind of webby in appearance. And again, the rough ER has all these little spots on it. Those are ribosomes. The smooth ER does not have that. All right, so what's your main function of the rough ER? Again, the rough ER manufactures secreted proteins, So those are proteins that cells are going to release into the extracellular fluid. So that might be, a lot of times those are hormones or signaling molecules that cells are sending to each other. They make those inside the cell and they've got to secrete them over to the outside. And if those are proteins, they're going to be made on the rough ER. The rough ER also makes any membrane proteins. Integral protein just means a protein that is stuck in the membrane. It's integral. It's an integral part of the cell membrane. Also, the rough end ER actually makes phospholipids. So cells have to make those. They have to replace ones that wear out over time. So they get made in the rough ER. All right, how about your smooth ER? What is it doing? It has different functions. It has different roles in different cell types. For example, in the liver, you've probably heard that your liver does a lot of detoxification for the body, like it helps inactivate things like alcohol, but it also inactivates and breaks down drugs that you take or toxic chemicals like pesticides that we get exposed to pretty frequently. The liver has to break down glycogen, which is, again, that's a starch. Glycogen, just chains of glucose sugars that are being stored inside the liver and also in your skeletal muscles for time periods when you need a quick burst of energy there. So in the liver, the smooth ER carries out some of those functions. Steroid type hormones are actually made in the smooth ER. Steroids are lipids that are derived from cholesterol molecules. In the intestinal tract, the cells that line the inside of the intestinal tract, the smooth ER there will be involved in absorbing, producing, and transporting fats that you've absorbed from your from the foods that you've taken in. And you'll see when we cover the muscular system, the smooth ER is involved in storing and releasing calcium ions, which, as it turns out, calcium is going to play a very important role for muscle contraction that we'll learn about in Unit 4. All right, what is the Golgi apparatus? Let's see if we can advance forward over here. All right, so on this diagram from your textbook, all right, so over here you've got some rough ER. So we were just talking about how the rough ER manufactures proteins that are going to wind up in cell membranes, or proteins that are going to be secreted or released from the cell. And the Golgi is like the next area along an assembly line for that process. So those proteins first get made over here, and then they get sent to an area called the Golgi apparatus, which is depicted here in green. And it's like a series of membrane covered pancakes almost. They have a very flat-like structure inside the cell. And what the Golgi apparatus does is it takes those proteins and it modifies them if they need to be modified or changed in any way. It really is like a factory that's working on these proteins. And then finally, the Golgi apparatus kind of acts as a post office. So it packages up these proteins and sends them to where they need to go. So when it packages them up in little vesicles, membrane-covered compartments. Remember, we were talking about vesicular transport, and it may send those over to some other organelle, like they're showing you here, if those proteins are going to stay inside the cell, or it may send those to the surface so that they can be secreted. That process is called exocytosis. Or in some cases, it will send these vesicles to the cell membrane, and you'll have these little proteins that stay there in the cell membrane where they're supposed to be. So all those things like those carrier and those channel proteins we were taking a look at when we went over plasma membrane transport, cells have to make those for themselves. They don't just magically appear there. So they go through this factory assembly line type process that starts in the rough ER, proceeds to the Golgi, and then the Golgi packages those proteins up in these little membrane covered bubbles or vesicles and sends those on up to the surface. So your Golgi is kind of like your post office for a cell. How about lysosomes? Lysosomes are membrane-covered organelles. Some people call these cell stomachs because they contain lots of digestive enzymes. And some of those are acidic, or they function in acidic conditions. Um, and many of those are hydrolases. They do hydrolysis chemical reactions where you add water to things like proteins and carbohydrates to break them down into their little building blocks, their monomers. Lysosomes in some cell types are involved in digesting bacteria and viruses and toxins. Remember, we talked about phagocytosis, where you might have a white blood cell that engulfs a bacterium or a virus and takes it in. Well, once that happens, then the lysosomes are going to dump all these digestive enzymes in there and break down that poor little bacterium or virus that was ingested. Lysosomes, these cell stomachs with all these digestive enzymes, they actually even have to break down worn out cell parts, non-functional organelles, so that they can be replaced. And then in some cases, what about your own tissues and cells when they die? Like all your red blood cells, when they die and have to be replaced, they only live a few months, and then they have to be broken down, and all of their little parts have to be recycled. Well, what's doing that inside your body? You have cells that do that. They actually take in your own dead and damaged cells and tissues, and they use all these digestive enzymes inside the lysosomes to break all of those different things down. Peroxisomes, that's another membrane-covered organelle inside cells, and those have enzymes in them that detoxify. They're also involved in detoxification. Your smooth ER also does some of that as well. One of the things they're really good at detoxifying are molecules called free radicals that usually have oxygen atoms in them. You may have heard about free radicals if you've ever woken up on the couch at 3 o'clock in the morning and there's an infomercial on, like for these creams and so forth that help ward off aging. A lot of times they'll mention free radicals. Free radicals are very destructive type molecules that we get exposed to that can damage your cells and tissues very easily. So your cells actually have to inactivate or neutralize those types of free radicals in order for you to be able to stay alive for your 70, 80, or 90 years that you'll hopefully live here on the earth. And so your peroxisomes have enzymes inside them that perform those functions. All right, cytoskeleton. We're not going to get too heavy duty into the cytoskeleton, but all right, got my pen up here. Inside a cell, you do have a framework of thread-like proteins and tubes that crisscross their way inside the cell, and some of those are called microtubules, some are microfilaments, some are intermediate filaments. Don't even worry about that. I just want you to know that the cytoskeleton basically provides is a meshwork of these protein threads or filaments. Filament means thread, and tubes that help provide a cell shape and structure, make it tough so that if it gets compressed, you've got a framework in there, almost like the skeleton of your body in a sense, that's preventing that cell from becoming damaged. Can also play a role for movement of cells, and they can even act as little highways for things like those vesicles we were taking a look at, how they're moving from, say, the Golgi to the cell surface. They don't do that randomly. They actually hop onto the cytoskeleton and slide where they need to go. All right, the centrosome. You know what? We're just going to bypass centrosome. Don't worry about that. Cellular extensions. You'll hear about these at times in anatomy and physiology. Cellular extensions can include some cells. Some cells have on their surfaces little tiny hair-like structures that are called cilia. And what those do is they whip back and forth, and they help move substances along a surface. For example, you've got the cells that line your trachea or your windpipe have little cilia on them, and what they do is they move and they sweep substances upward out of your windpipe and back up towards your pharynx, the back of your throat. That way, dirt and germs can't make their way down deep into your bronchi and your lungs where they're not supposed to be. Flagella on a sperm cell, the tail, that is called a flagellum. Flagella is plural, and that tail whips back and forth to allow the sperm cell to move. They're actually built from microtubules. Those are protein tubes, which you also have as part of your cytoskeleton, and that's why that's mentioned there. Motor molecules are special proteins that harness the energy that's needed to allow these cilia and flagella to actually move and whip back and forth. All right, finally, what about the nucleus? So those were all the structures there in the cytoplasm. What about our nucleus? Here. Let's see. Well, here we go. Here's our nucleus. Nucleus tends to occupy a lot of space inside the cell, and here we've got it cut open. And on the inside, you have a lot of chromatin in there. That's another name for DNA wrapped in proteins. Your DNA molecules are, if you took all of the DNA out of one of your cells, it would be several feet in length, believe it or not, if you could extend it all the way outward. So all that DNA has to be wound around proteins to keep it compressed and packaged there inside the nucleus. So the DNA protein combination that compresses the DNA, all that genetic material, small enough and tight enough so that it can fit inside the nucleus, that's referred to as chromatin. Let me back up here for a second. Yeah, a couple of terms over here for you guys to be familiar with. Most cells in your body are uninucleate. That means they have one nucleus per cell. Your red blood cells are unusual in that they are anucleate. A, that prefix there, means without. So those cells actually have no nucleus. And then there are a few cell types in your body that are multinucleate that we'll hear about as you go through your A&P classes, which means one cell has more than one nucleus. Those are more unusual. And here's a depiction of what DNA actually looks like inside the nuclei of your cells. So here's your DNA double helix over here. And so again, that has to be very tightly compacted to fit inside the nucleus. And so it gets wrapped around these little blue discs that you see here. Those are proteins that are also called histones. You don't have to worry about that. But the DNA gets wrapped around these proteins and coiled up really, really tightly so that it can fit inside the nucleus. So the DNA protein combination there can be called chromatin. I'm sure you've heard of chromosome before. The DNA in the nuclei of your cells is divided up into different DNA molecules that are called chromosomes. And you have 46 chromosomes in most of your cells. And you got one set of 23 from your mom and one set of 23 from your dad when sperm met egg. And sometimes that term chromosome is used to describe, if you've ever seen pictures of chromosomes depicted like this in this kind of X type shape, that is super condensed down, tightly coiled DNA wrapped around proteins. And when you see that X-shaped depiction like that, what's happened there is the DNA has actually been copied. It's been replicated. You now have two copies of the DNA molecule, and that cell is getting ready to divide, and those two copies are going to split apart, and one's going to go to one daughter cell, and the other copy is going to go over to the other daughter cell. So that term chromosome, um, originally it was used to really refer to DNA molecules that are really super tightly compacted down right before a cell is going to divide. Today that term is really used more loosely now that we're beginning to understand a whole lot more about all these different DNA molecules that we have inside our cell and what genes are located on them. We know the AGCT sequences of these DNA molecules and where all the different genes are located, and we're beginning to figure out what those genes do and so forth. And so a lot of times people today just refer to chromosome number 1 or chromosome number 3 or chromosome number 22, and they're not necessarily in that case thinking about DNA that looks like this in a cell that's getting ready to divide. That term chromosome is used a little bit differently today than it used to be used in the past. All right, so that concludes our little overview on the cell structures, parts inside of the cell. And I don't want you guys to get too stressed out over this topic. I know there's quite a bit of information there. We just want to have a little bit of an overview so you are beginning to get familiar with those different cell structures for when you hear about them in the future. And what I would like you guys to do, though, is use the practice questions or your homework questions in Mastering A&P. And I'll also have some additional practice review questions posted for you guys on Blackboard, and that should give you some idea of the level of detail that you need to know the material from this particular video lecture. Also use your information in your study guide for this part of Unit 1. All right, well, you're done with all of the video lectures for Unit 1, and I know you're really upset about that, but please practice a lot before you go on to take the exams for this part of the course. And let me know if you have any questions. I'm always here to help you.